Alright guys, let's go over the homework guys. If you have any questions. I think the last four, those were FRQs, so I'll go over those guys, all of them. But let me know guys, the multiple choice sector, which is the first 35 questions guys, go let me know if you have any questions, just call them out. I'll go over those questions for the first 35. And again, like bring your devices, because I'm not handing out you guys papers, okay? Alright, questions, let me know guys for multiple choice. Any questions? Any questions? Let me know. Questions? First 35, multiple choice. Let me know. Some of you guys got like everything right, so but let me know. Questions? No questions? I saw some of you guys did like pretty bad, just so you know. Let me know. Just give me numbers.
I got some wrong, but I just want to know, is there a difference between ADP and AOP?
The typo.
Oh, okay,
Yeah.
so it's
Yeah, ADP, yeah, it's a typo. I forgot which number that was.
It was one of the first.
Yeah, it's AOP. They made a mistake there. Yeah, this is a really old AP exam. I think like that question, and then I think there's a typo there, like 1986 or something.
Oh, okay.
Any questions from you guys? Numbers, guys. Like, don't be shy and give the numbers. Some of you guys didn't do it, and I tell you, man, I don't know. Questions, concerns? Let me know. Homework? No questions, guys? Still about us? I should be asking you questions then, so you guys got everything right. Any questions? I think, like, I don't know. Some of them, as I figured, guys might let me able to solve it. I don't know. Like number 35, I don't know if you guys knew what the question was, but please, guys, ask appropriate questions if you need it.
I'm still trying to connect to the Wi-Fi.
Huh?
I'm still trying to connect to the Wi-Fi.
The password is right in front of you. No questions, guys? No questions? And please make sure you guys bring your devices, alright? Guys, otherwise I'm gonna go to the FRQs, guys. So if you guys, I'm not gonna force any questions from you guys. Alright, guys. I mean, this is number 36, 37, 38, 39. Those are the last four FRQs, guys. I mean, if you guys have anything else, just let me stop me. Alright. Alright, scroll to guys 36. Okay, so I forgot which year this was. Maybe 2017, 16. I forgot. This was the actual FRQ question number 1. So number 1 and number 2 for the AP Bio FRQs, they're the ones that are worth like a lot of points. Okay, so you always have to kind of watch out for number 1 and 2 for the FRQs every time. Okay, and a lot of these questions have been used. I think like for number 38. I mean, I'm for sure, guys. If you guys go to school, like Jericho folks, I mean, this is one of your school exams, by the way. So pay attention to these questions, please. So you guys read the question for number 1. I'm not gonna repeat it. Okay, so graphing, I'm not interested in graphing right now, guys. I mean, you guys can figure out the graphing stuff, the labeling and all, guys. I mean, that's something you can figure it out. But the question is, guys, okay? So, do you see this, guys, like absorbency, like you see the number 389? So, this number right there represents, guys, the wavelength, okay? The wavelength is equal to 389 nanometers. I think that's what they're referring to here. Okay, I don't know if they mentioned it, I'm not sure, guys. Yeah, I think they did. Absorbency of the absorbency of the 389 nanometers light. So I don't know how much you guys remember, guys. I mean, so with these things, I mean, absorbency, that's more of an AP Chem perspective, in terms of like, okay, so what kind of light gets absorbed and all. But if you look at it, guys, I mean, the wavelength of 389 absorbed. So this is around what, guys? This is blue. Okay, this is blue color, in terms of 389. So, it's something we'll discuss a little bit, again, a little more in photosynthesis, right? So, all spectrum light you know, which is like from like blue to red, okay, or violet to red, whatever. So, you know, the range is around like what, like 300 nanometers, like, you know, to like 700 nanometers. That's around the range of visible light spectrum in terms of wavelength. So, you know, so they're specifically looking at one of them, which is 389. So you got to understand. I mean, not that this is a question based on that, just so you know, but they're saying whatever this thing is, it's the color turns brown. So keep in mind, guys, here. Okay, so those are coming from the AP Chem. You guys probably know this pretty well. Again, like I'm not like very artistic kind of person, guys, by the way. But brown is what, like somewhere here, red and like yellow, you mix them, like what, like it turns into like brown or something like that. Yeah, I don't know. So. So the reason is because, I mean, so out of all the spectrum lights, guys, if they absorb somewhere on the blue spectrum or violet, I mean, the color comes out to be like more like brown. So that's why they set the absorbency as 389, because that's the light wavelength that gets absorbed, you know, by the product. In this case, it'll be benzoquinone. That's a ketone, guys. You know, so it's an alcohol, which is catechol plus the oxygen turns into what? Turns into a ketone. So this is one of the things, guys. I mean, so it's good to know. Okay, where if you have the alcohol, okay, this is like some of the things that you'll never hear from anywhere else, guys. This is not even AP Chem, because this is Olympia level, like higher level, guys. By the way, like chemistry perspective. So when alcohol gets oxidized, so whatever you think the term oxidation means, guys. By the way, when alcohol gets oxidized, okay, it either turns into aldehyde or ketone. Okay, aldehyde. So you guys should know what aldehyde is right now, guys, because yeah, you guys know the functional groups. Or ketone, okay, depends. So that's one of the things. So alcohol gets oxidized means what, guys? Yeah, like either you mix with the oxygen, or you take the oxygen away from or electron away from the alcohol, so it turns into the aldehyde or ketone. Okay. In this case, if you look at it, I mean, in catechol, you put the oxygen there, and then what? It turns into a ketone. If you look at it from the name, guys, benzoquinone, that's a ketone. Okay. So it's good to know, guys. And again, so if you coming from a very heavy chemistry background, then you probably will know what the hell is happening here. So I mean, not that that's really needed. You know, not that you have to understand A to Z, you know, for the entire process of what the question is asking. But what they're saying is, once the product has created for the benzoquinone, what happens, guys? It's a color browning. So the product, which is a ketone, is sort of what, guys, absorbs the wavelength of this. And you know, the product, you know, whatever that's surrounding the product appears to be brown color. That's the understanding of this, okay? And the question is asking, guys, A, like, you know, like this, the graph. I'm not interested in that for now. Part B is asking, guys, based on experimental results, estimate the optimal pH. I mean, it's like, I mean, dude, like, unless you're, I don't know, not there, man. You know, so optimal pH is what? If the enzyme, the polyphenol oxidase, if it works really well, what happens, guys? I mean, yeah, dude, like the degree of the change will be greater, right? So if you think about that, guys, okay, so the change in the absorbency, that's the biggest one as well, 0.83, okay? So I don't know if you recall these things, guys, by the way, the equations for the absorbency and the concentration and stuff like that, guys. What's the equation, guys? This is called the Beer's law, right? So you're not like, I don't know. There's a dependency on the equation. Please come in time. There's Beer's law, right, which is absorbency is equal to what, guys? A times B times what? See, that's the equation. So as you can see, guys, there's a reliance on the AP Chem stuff, okay, on AP Bio, right? So absorbency, how much gets absorbed, and what? Concentration, which is amount of benzoquinone. So if it means there's a greater absorbency, that means what? There's a greater amount of benzoquinone. Okay, that's what it means, right? So if you look at the test tubes, right, at the variance of pH, so you see the greatest amount of change, or the highest concentration, or greater absorbency, when the pH is around 0.83, or I'm sorry, pH is around pH is around 7. When pH is around 7, okay, the absorbency is the highest value. Okay, what does it mean, guys? Yeah, you have the highest concentration, right? So I think that's what the question is looking for, guys, part C, and then you have to kind of prove them, like, okay, it's how you got into that, you know, how you get to that result, right? So again, like, I understand that this has nothing to do with AP Chem, okay, but there's a reliance on AP Chem stuff, okay? It's good to know if you have more information, okay? Not that it's impossible to solve if you don't know what the hell this means, but you can kind of guess, right? But at the same time, if you have a background in Beer's law and AP Chem stuff, that'd be really nice, okay? Any questions, guys? Let me know on part B. Questions? Let me know. You guys need extra explanation for this one now. And part C, the question is asking you guys, so in AP AP Bio, I'm sorry, the questions about control is a big deal. They ask a lot about control stuff. Okay? A lot about control. Okay? So you gotta understand the core of, I guess, the experiment, guys, by the way. So when you're doing experiments, right? So basically, guys, you have independent variable here, and you have a dependent variable, right? So you know, independent and dependent variables here. So whatever the independent variable is, it impacts the results. Okay, so you know, in this case, independent variable is what? pH, right? And this is what? Absorbency. Do you understand, guys? So that's basically, guys, the core of the experiment is they're changing the pH, okay, which will impact the absorbency. That's the core of the experiment, okay? But now, what you have to understand is this. Will there be any other factor that can what? Change the absorbency, okay? So are there any other variable that can change the absorbency? Because that's like basically what you have to think about. So that's why you need to have something called control, okay? So you just want pH to impact the absorbency. Okay, so you gotta do some, you know, like have a control in the back, so that what? Okay, so it is the pH that's changing the absorbency. Okay, so this is a big deal for the AP. Okay, and not for the Olympiad guys. For they don't bring up control, I think, at all for the Olympiad at all, actually. Okay, so this is like college board stuff. Okay, so let's look at the equation, guys. Okay, so they got a catechol, which is the alcohol, plus O2. And there's an enzyme, okay, which turns into like benzoquinone, okay, like I'm gonna say B plus H2O, okay. And now the expectation is, okay, the pH will change. The pH will change the activity of the enzyme, okay, which will result in what, guys? Yeah, impact the production of the product, okay. But okay, so what are the control, guys? So what do you have to think about? Okay, so what's your, you know, what do you have to really look at? Okay, so is it the pH and enzyme only that impacts the absorbency, which is the amount of product? So you got a question, right? Okay, so what about these guys? Can pH somehow impact these guys, the reactants? Like, okay, so for example, guys, okay, so when pH goes low, okay, like whatever the reaction can result in what? Change of the catechol, which means what? Nothing gets created, or something like that, right? So, you know, so pH might not impact the enzyme, but what? It might impact what? Catechol. That's what I'm saying, guys, here. So that's what? That's like additional variable. Okay, so we're thinking pH has an impact on the enzyme, and okay, so that's an impact on the reaction overall. But can pH also impact catechol? Okay, that's dude. Isn't that like a thing you have to think about? So then, what kind of experiment can you set up for this? Okay, well, what are you gonna do? Yeah, so the control experiment should give up, guys. Okay, remove the enzyme. Okay, remove the enzyme and what? Run the same process, okay? Run the exact same experiment. You understand what I'm saying? Okay, then what happens then? Yeah, if you change the pH, okay, and then yeah, measure the same thing, okay? So if it does also shows a variation, then what happens, guys? Yeah, dude, this might have an impact on the experiment, okay? But if you change the pH without the enzyme, and this is a very little variation in the absorbency, okay, what happens then? Yeah, this doesn't have impact at all, okay? That's what you call the control testing, okay? Which is what? Just to make sure that there are no other variables that impacts the resulting, okay? That's basically what it is. And now there is, so whatever I mentioned, guys, right? The experiment of this and stuff like that. So without the enzyme, you know the product is not going to really happen in bio, as we discussed, right? This kind of testing is called negative testing, okay? Like negative control testing, okay? So there's a positive and negative. Okay, so negative control is what? You know for a fact, okay, so you know for a fact, okay, you're not gonna get a product, okay, for example. So without the enzyme, what's the chance that we get a product? Very little, okay? So that's an example of what they call negative testing in AP Bio. And I don't know, guys, like past year or two, you know, they never asked about whether you need to know negative or positive, like control, but now they're asking, like you have to know the difference between negative and positive. And positive is what, guys? You know for sure things gonna happen. Okay, you know for sure the product will be created, okay? So So those are what they call, guys, of negative positive testing. So basically, the correct answer here would be what? Okay, so run the same experiment, okay, without the enzyme. Okay, that could be one of the that could be what one of the you know, the control, negative control testing. Or you can say, well, okay, run the same experiment without catechol, or run the same thing without the O2, or something like that, okay? Which might reduce the number of variables in a reaction. So, an experiment perspective. That's what they call a control. You have to understand why they're doing this. Any questions? Let me know, guys. So I haven't really gotten into your work individually and then saw what you guys wrote. I didn't read that, but this is the core of the control testing and such, okay? Any questions? And in part, these outside of your, it's something that you guys can't really answer right now, okay? So this is questions asking like genetic engineering can be used to disrupt the specific gene, blah, 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 blah. So describe the browning apple would be affected, the strain of the apple genetically engineered to lack the functional copy. So I mean, yeah, if there's no enzyme, it's just not gonna happen, okay? Or very little browning per se, okay? So like, but this is outside the scope for us, guys. I mean, so I don't think we have to really discuss right now. Okay, any questions? Let me know, guys. Number one. Questions? Let me know.
37.
Huh?
37 or number 8.
Yeah. I'll go over all that part of you. Okay. And let's take a look at, guys, 37. The next question, which is number 8. I forgot which year this was. And this is where I think we sort of left off last week, like the Michaelis, like Menten equation and stuff like that, guys. Okay, if you recall. So, if you look at this, guys, the graph. Let's look at the graph, guys. So keep in mind, guys, here, by the way. So I think I saw this too yesterday, okay? So I went in and solved it online and stuff just to check the answers and all. So just to let you know, right? This represents, you gotta be very careful, guys, here. This represents what, guys? The rate of the reaction, which is like, we call that V, right? Remember that, guys? This is the amount of substrate, okay? Substrate concentration. We talked about it, guys. I mean, so the reason I mentioned that, guys, is because on multiple choice questions, they had this as the total amount of product. Okay, that's completely different, like, graph. So one of the multiple choice questions, guys, they had, this is the amount of product produced, okay, versus like whatever time, I forgot. So that's what we got. Okay, so this is not the same graph. This is the rate of the reaction versus the amount of substrate. Okay, that's the Michaelis-Menten equation, the graph. Okay, but this is completely different, guys. Okay. So question is asking, guys, here, and I think they represent, guys, like they didn't really. I mean, the AP Bio, like, it's not a big deal, guys, here. So this is what we call the Km. Okay, this is like the half of V, the V max over 2. You know, so this is basically, I don't know. We don't have to really go too far into this for AP Bio. So the question is asking, guys, if there is a question, right? As you can see, if there's a question about, like questions like this, it's always they ask you about the tertiary structure and protein and all, how that's impacted and all. So the question is asking, guys, connect the primary structure of the enzyme to the overall shape. So, dude, like when they say overall shape, right? What are they referring to, guys? What are they referring to, guys? This is what? Tertiary. Typically tertiary or quaternary shape. Okay, so like the protein folding and all that stuff, guys, by the way. So what is the primary structure of the protein, guys? What is that, guys? A sequence of the amino acid, okay? So again, methionine, like, lysine, glycine, whatever. So the sequence, right? So question is that, so different way of asking, how can sequence, which is the primary structure of the protein, impact what? Tertiary or quaternary, okay? So you guys should really know that, guys, at this point, guys. You have to explain it, guys, where, okay, so tertiary structure is what, guys? Tertiary structure created by what? Hydrophobic interactions and what? Like hydrogen bonding, disulfide bridges, and stuff like that, guys. Ionic interactions of what? The side chain groups of the amino acids. Okay? So based on a specific sequence, okay, they create what? Specific shape. Unique shape arises from what? Interaction of side chains. Okay? Because the sequence of the protein is different, that's why what? They have a different what? protein folding in what different tertiary structures, okay? And that's basically what you have to mention, guys, is to give them like step-by-step explanation how a primary structure turns into a tertiary, okay? And yeah, that's overall thing. You gotta be very like detailed with the explanation for part A, okay? One of the things to understand, guys, by the way, is I don't know, like your school teacher might be like, they might say, oh yeah, you write a response, like, yeah, good job, like 100, on your test. But that 100, guys, might not result in the actual AP exam. You gotta be much more detailed, and you can't make mistakes, guys, on the explanation on the AP exam or the FRQ sector, okay? In part B, the question is asking us predict the effect of adding a non-competitive inhibitor. So this is one of the stuff that we talked about, guys. So what do you guys know about the non-competitive inhibitor? What do you guys know about that, guys? I mean, so I think we talked about it a little bit towards last week. So one thing is what? So there are, I think, 3 inhibitors that we talked about, right? So one is what, guys? Irreversible. Okay. Irreversible inhibitor. Okay. So what the hell is that, guys? What's irreversible inhibitor? So if this is the enzyme, right? This is the enzyme. Okay. And then again, keep in mind, guys, here that's the active site. Okay. This is active site, right here. Typically, irreversible inhibitor is what? It binds itself to the active site. Okay. And what? And irreversible. Like this is the thing, right? In AP Bio, irreversible means what, guys? So this is strange, but they're saying it's covalently bond. Covalent bond. Again, there's something about AP Bio, guys. They think covalent bond is very strong. Okay, so this inhibitor binds covalently to the enzyme on or near the active site and what? Complete blocks it. Okay, so typically, guys, again, covalent bond is what? Like a permanent change for the enzyme. So which means what? Yeah, they can't take this back out. It's game over. Okay, so that's what they call, guys, irreversible, right? And we also talked about competitive. Okay, competitive inhibition, right? So what is competitive inhibition, guys, by the way? So what the hell is that, guys? So competitive inhibition is if this is the substrate, okay? But now, if you put an inhibitor, okay, around it, so what happens, guys? Yeah, they will compete for the active site, okay? So, dude, think about it, guys, okay? If you guys have 50 of these substrates and 100 of these inhibitors, okay, by chance, what happens, guys? Okay, 2/3 of the time, it's what? The competitive inhibitor will bind to the active site. So which means what? Will slow down, okay, the substrate binding to the active site. That's what we call competitive inhibition, right? Just so you know. So, but you gotta understand, guys, in terms of competitive inhibition, okay, then what happens, guys? What do you think? So look at this graph, right? At a lower level, so yeah, you gotta be very understanding with the graphs, right? Like by perspective. Whenever they give you a question like this, substrate becomes the independent, the rate is a dependent of the substrate, okay? Typically what, guys? Nothing else changes. You know what I'm saying, guys? Nothing else can change. There's no other variable, which means what? The amount of the inhibitor doesn't change, amount of the enzyme does not change. You understand, guys, what I'm saying? Okay? So if you increase the amount of substrate, what happens, guys? Like, dude, if this is what, guys? Like 10,000. You increase. Okay? Think about what will happen. What do you think? The amount of inhibitors you have is what? Might be what? Like, dude, negligible. That's the way. This is what, guys? 1%. Okay? So what happens, guys? 99% of the time, substrate will bind to this, and only 1% this guys will bind to that guy. You know what I'm saying? So which means what? As this gets higher, as the amount of substrate increases, as the graph goes, is what happens, guys? It makes no difference. Okay? So which means what? Okay, so maybe on the bottom it does have an impact. What happens, guys? Like it'll be more like this. Okay, so as the substrate level goes high, what happens, guys? Yeah, it goes to the same. Okay, so that's what you kind of see from what? Competitive inhibition, right? Then what is a non-competitive inhibition, right? What the hell is that, guys? So Non-competitive inhibition is what, guys? The inhibitor is binding to a site of the enzyme other than its active site. Okay, so it's gonna bind itself to somewhere else, okay, other than the active site. And what happens, guys? If you bind here, what happens, guys? The enzyme shape will change, okay, slightly. So basically, what happens, guys? Yeah, like the things will be a little different, guys. So basically, guys, the shape of the enzyme now will change just a little bit. It widens out a little bit, okay? So now, for example, okay, what happens, guys? Substrate can still bind to this active site, okay? But what happens, guys? The reaction might be a little slower. It doesn't do a good job of what? Catalyzing the process, okay? So, which means, guys, overall, it might lower the maximum rate, okay? So it might slow it down, something like this, okay? So this is what they call competitive inhibition. So competitive inhibition is not same as allosteric inhibition. It's completely, like, they're different, okay? They're not the same thing, okay? So although the similarity is what? They're binding to a site other than the active site, okay? But the allosteric inhibition is a little bit different, okay? So, keep that in mind, okay? So that's what competitive inhibition is, okay, by the way, okay? So, like, yeah, things can still happen, okay? So this binding to other than active site, it can still, like, yeah, make it happen, but at the same time, it might not be at the optimal for the speed of the reaction. Okay, so we talked about, guys, what these inhibitors, how the enzyme assists of the lowering the activation energy, right? Like providing the microenvironment and providing what, guys, like template for the substrate to go in and what, the react and such like that, right? So the question again is asking, guys, predict the effect of adding the non-competitive inhibitor, the reaction mixture, on the rate of the reaction at high substrate concentration, which means what? It'll be lower, okay? With some of the stuff that we talked about. And then support your prediction by describing the non-competitive structure. It changes, okay? So just like how we talked about, guys, I'm not going to repeat myself. So that's basically how it occurs. I mean, it'd be nice, okay, so if you read what I provided, guys, okay? It doesn't change the Km, okay? That's one of the stuff that we talked about, I think. And also what, guys? But it lowers the Vmax, okay? That's one of the good things to note. But again, like, I think they don't want to talk about the Km portion, okay? Those are only for, like, the Olympus stuff, okay? All right, so let's go to, guys, number 38. 38 guys are 39. So this is a cyanic acid, by the way, HCN. HCN is cyanic acid. And this question, guys, I'm sure this I think the past like couple of years, like the kids from you guys, like they asked this question actually. This is one of the questions that they had on the test actually for biochem. And then the question is asking you guys, right? Based on the chemical structure of the cyanide, identify one type of biological macromolecule that could serve as a chemical precursor. So they got a what? Question is asking, guys, this thing has a hydrogen, carbon, and nitrogen, right? And the question is asking, guys, okay, so where could have possibly this came from? So what kind of macromolecule has hydrogen, carbon, and nitrogen? Okay, so the big part is what? This guy right there. Okay. So you should understand, guys, carbohydrates, right? Carbs, they have what? Typically, they have a carbon, hydrogen, and oxygen. Okay. Lipids, typically, okay, they have what, guys? Carbon, hydrogen, and oxygen. This is a terrible question, by the way. Proteins, what do they have, guys? Carbon, hydrogen, and oxygen, and what? Nitrogen and sulfur. That's what they got. All right. What about the DNAs and, like, nucleic acids? What about them, guys? They have a carbon, hydrogen, and oxygen, phosphate, okay, and nitrogen. So the question is asking, guys, okay, which one has it all? Okay, which is what can be the answer, guys? Either protein or DNA, one or the other. Okay, this is where they could have came from. So these two typically don't have the nitrogen there, right? In part B, the question is asking, guys, it's having proposed that apple trees are, yeah, it's not for you guys right now. Part B, guys, I mean that's for way down the road, guys. Okay, ecology. Let's go over 39. Okay, so this is an interesting question, by the way. 39. So, if you guys I don't know, guys. If you guys, I don't know where you guys are at, guys, and if you guys have been on a road trip and stuff like that, by the way. But if you guys drive to near Ohio, Iowa, those places, guys, dude, I tell you, man, you drive like few hours through cornfields, Okay, like dude, like just, you know, there are these like cornfields, guys, and then you can't get out of it. Like the roads, guys, like left and right, like cornfields, you guys, you know. And I tell you, man, like my friend actually, okay, he got a PhD in economics, and in Iowa, like State University or something, and then you know, he told me that. I mean, so one of the funny things that he told me was, like corns, like when they harvest the corn, okay. I mean, he told me that when they harvest it, they heat up. They heat them up, okay? Like they heat it up and like before it gets sold, like I don't know what temperature they heat it up to, but they said they heat it up and package it out and they sell it to people. So and you can kind of understand, okay, why they're heating up the corn after they get the harvest the corn, right? If you look at this, okay, so the corns, they have the content as well, guys. Like they have it as sucrose. Sucrose is where we can taste the sweetness, okay? Do you understand, guys? Like sucrose and glucose, that's something you can taste the sweetness, okay? Because that's like the table sugar, okay? But now what happens, what, guys? Okay, so as time goes by, okay, plants are, you know, they're what, guys? Typically, when they lack a resource, they convert the sucrose into what, guys? Starch. Starch is how they store their food, right? So if you leave the corn itself for a long time, just let it sit there, what happens, guys? Yeah, they will turn the sugar into a starch. You can't taste the sweetness, it turns into a starch, by the way. Guys, you know, you like starch, try, guys, eat starch. You can't feel the sweetness there, okay? Yeah, they heat it up and what? They just leave it as a sucrose, okay? So I think like that's where they get this question, okay? The main idea of the question, guys, number 7. And the question is asking guys, describe the temperature above 35. So again, like if temperature goes above 35, it's hard to say, man. Like 35, guys, if temperature goes like right above 35, it's hard to say it denatures, okay? So denaturation, guys, denaturation occurs at a much higher temperature, okay? Like, okay, so the optimal like temperature for starch synthetase 27 to 30, you know, 30 to 35 is a sucrose synthase. So, oh yeah, if you put it at 36, will they denature? Very unlikely, okay? Denaturation doesn't occur at temperature 36, okay? So, if you bring it up to, for example, guys, like maybe like 100 degrees Celsius, yes, it will start to denature permanently, right? So, you know, well, so it's hard to say, but if you mention denaturation, okay, they're not gonna mark you wrong, okay? So, you know, when the AP, okay, that's fine. But I think, guys, just thinking about it, guys, okay, yeah, just above 35, I mean, it might not be nature, but what? It can change the structure of the protein slightly, okay, which will decrease the function of the protein, okay? Maybe that's a better understanding, right? So the question is asking how temperature above 35 most likely impact and affect the structure and function of the starch synthesis. I mean, so you could, if you could have said denaturation, that could have been the correct answer, okay? That's what the answer said, it's fine. But you could have also said, well, yeah, temperature 35 might have an impact on some of the hydrophobic interactions, hydrophilic interaction, blah, 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 and what maybe change the configuration or conformation of the protein slightly, which will decrease a function of what? Turning a glucose into a starch. You can say that as well. And then the question is also saying, guys, use the information to provide predict the most likely consequence of the starch content. If it's temperature 33, guys, then it's above the optimal temperature for starch enzyme, okay, which means what? The more likely it will stay what? Sucrose or glucose, okay? Because like this one, temperature is what? Like 30 at max. So more likely, guys, will stay at the sucrose or glucose. That's probably what the answer is. I mean, it's a very simple question because you think about it, right? If you know what you're doing, guys, it's really not that bad for the AP question that FRQs. But believe it or not, man, there's a lot of things messed it up. 100%, guys, okay? Any questions, anyways? Homework, guys, let me know. Multiple choice. Any questions, concerns? I looked at all these yesterday and I checked off all the answers, and usually if I get everything right, I don't even like to mention too much. There's
Could
some.
we go over question 2?
Huh?
Could we go over question 2?
Number 2? That's a good question, actually. This is a good question, actually. So substrate molecule may be bound to the active site. So this is one of the stuff that I think we talked about, right? So the enzyme binding to the substrate, like what kind of interaction are they seeing? So keep in mind, for bio, right? like the way they make it sound like is that when they create a covalent bond, it's permanent. So like I don't know why. Okay, so if this is the enzyme, okay, and if this is a substrate, okay, so like okay, so we understand it goes in and like it does its magic and stuff like that, right? So okay, so what kind of interaction does it have with this? Basically, what will happen is what they have everything except for the covalent bond. Okay, that's basically what it is. You can't have a covalent bond with this guy and that guy. Then what? That's like more of an irreversible like inhibitor kind of thing. So it could be hydrophobic hydrophilic interactions, and it could be ionic as well, hydrogen bonding, but it cannot be a covalent bond. Okay, so but kind of so that's one other thing, right? If you read a textbook, okay, there's a textbook called Chempill, right? They say, okay, sometimes, okay, rarely, substrate can bind to this guy with a bond. Okay, so that sort of also provides like catalytic process as well, but rarely it does. Okay, so I think that's one of the things you have to look at. The question is asking, okay, so except for what, guys? I think peptide bond right there. Okay, peptide bond is what? That's a covalent bond. Okay, so as we know, carbonyl group and nitrogen, that right there is covalent bond. Okay, so that is unlikely to happen. Okay, good question. Anything else? Let me know. Any other questions, guys? Let me know on the homework.
Could we do the 3?
Huh?
Could we do the one right after 3?
No, what after 3? Number 4?
No, number 3.
Number 3.
The one after the one we just
I thought, okay. So what is this, guys, by the way? This is what, guys? If you look at this, I told you guys you don't have to draw this. If you see it, you have to know what it is. Yeah, what do you think? What do you think that is? What is this?
Nucleotide.
Nucleotide. So we had a conversation about this, right? When they say a nucleotide, okay, so very simple definition is what? Like, so I'm not going to do the whole thing here. So the simple definition is of nucleotide is what? If you have a nitrogenous base, okay, carbon, like a sugar molecule, and one phosphate, this is what they call a nucleotide. Okay, so but they got like what, 2 more? Actually, no. So you can call this. I mean, I don't like it being called a nucleotide. Okay, by the way. So I don't like it. Okay, so rather than that, okay. So it's got a nitrogenous base, okay, and it's got the sugar. Okay, this is what do we call? Yeah, remember, guys? What do we call from here to here again, guys? What do we call this? Yeah.
Nucleoside?
Exactly, it's called nucleoside. Okay? And it's got what? It's got what, guys? The 3 phosphates. Okay, so yeah, so this is what, guys? It's called adenosine. Okay, adenosine. Okay? So that's a nucleoside. Like guanosine, right? And what? 3 phosphate. Okay? Like basically, guys, I mean adenosine triphosphate. Okay, this is what? This is ATP. This guy's ATP, by the way, okay? So we know. So because it's got 3 of those phosphate groups and what? Adenosine, okay? So we like adenosine triphosphate, okay? So question is asking, could be any of those following except, okay? I mean, they said nucleotide there, okay? So monomer used as a component of the polypeptide, so I don't think so. It is actually, because DNA is polypeptide, right?
Yeah.
So I think, reactant used to, I think the best answer is, I'm sorry.
The answer is B.
Answer is what?
Answer
Yeah,
is
they
B.
say the answer is B.
But
I'm sorry, polypeptide, I'm sorry. Polypeptide is what? It's a protein, my bad. Yeah, B is the answer. You're right. Yeah, protein. So I thought.
That's so obvious.
Yeah, I thought they said polymer. My bad.
Yeah.
So
Me too. I read polymer.
Yeah. So the answer is more likely. Yeah, B is the answer. Opposite answer. Yeah, that's the answer. And then. So reactant used to synthesize of nucleic acid. So. Yeah, yes. So basically, this is a question maybe for a different time, guys. By the way. So when they are like producing like DNA, deoxyribonucleic acids, okay? So I mean, again, first thing first, guys, by the way, as you can see from the diagram, this is the OH group, right? If there's an OH group, we have what? What do you think this is what? The second carbon is an OH group, which means what? This is what? This is ribose. Which means what? This will produce RNA. Exactly, right? So, but keep in mind, guys, here, by the way, whenever the DNA or the RNA process, right, synthesis, right? So let's say, for example, guys, like DNA, you know, like, for example, replication of DNA, okay? So they got, like, you know, in terms of DNA replication process, they got a template and they're trying to match the other template, right? So what happens is, okay, D-ATP, okay, which means, guys, like deoxy-ATP, okay, like they're missing one of these guys. So it starts off with this, okay, and then they remove what? They remove the 2 phosphates, okay? That's called a pyrophosphate, by the way, and then they put it there. So ATP, guys, okay, so DATP or ATP, okay, or like GTP, like CTP. So those are all what, guys, like being used as what? Synthesis of the nucleic acid. So that's the reason why, guys, by the way, you know, like reactants used to synthesis of nucleic acid, okay? Reactants, okay? So this is a reactant, yes, okay? So it doesn't act as like a coupled reaction, you know, like providing the energy, but it is itself, okay? After they take off the pyrophosphate, and it itself being what? Used to synthesize the DNA strand, okay? So yeah, so it's an interesting question. And you gotta know, guys, also the term anabolic versus catabolic, okay? So what's anabolic activity? Is what anabolic is what, guys? Is a reaction where they build up a larger molecule from the smaller one, okay? And a catabolic is what, guys? You break down the large from the small, like large molecule into smaller molecules. That's what catabolic and anabolic process is, by the way, okay? And they said nucleotide. I don't like it, okay? So they said this is a nucleotide, guys. Hard to say, but for sure, B is a better answer. Okay. Anything else, guys? Let me know. Any questions, concerns? Multiple choice, let me know. You guys got a lot of questions wrong, but I'm not sure, man. Like, you guys, make sure you guys understand, man. Anything else? Let me know.
I got number 4 right, but can you just go over cyclic AMP?
Say again?
Can you go over cyclic AMP? It's in number 4, but I
A and B? Cyclic AMP?
Yeah.
So, so when you look at, right, AMP, what do you think it is? AMP? So we just talked about ATP, right?
Monophosphate?
Exactly, we're talking about mono.
Yeah.
Or monophosphate, right? So which means what? Like, dude, you remove 2 phosphates, okay, what happens then? This is AMP, okay?
Oh, we just talked about that.
Yeah, exactly.
Okay.
And then, like, okay, you gotta understand, right? Yeah. Cyclic AMP. So, again, like, dude, I don't know your background, guys, by the way. Cyclic AMP, okay? What is it being used for, guys, a lot of times? This is what? So, cyclic AMP is what, guys? I mentioned this in class. It's a signal molecule, okay? Typically used as what, guys? A secondary messenger, okay? For a lot of the cell communication process, okay? So, yeah, I'm not gonna go too far right now, okay? It's time. But again, like, but what the hell is that C? What the hell is that C, guys? I explained this. What is that C? Cyclic versus just AMP. What is the difference, guys? So that's AMP right there, okay? And keep in mind, guys, okay? If it's missing the oxygen, what happens, guys? We call this what? DAMP, okay? Deoxy. You know what I'm saying, guys? So you gotta understand, like, I mean, again, like AP, who knows, guys? Like, your teacher, unless they're definitely not Jericho, the teacher is absolutely crazy, you know, like the Columbia Prep or something, and yeah, like, DAMP, you gotta, like, unless something like that, you don't have to know it, it's fine, okay? Like, who knows? Your teacher might not bring it up, but cyclic means what, guys? So, cyclic means the phosphate group, okay? Like, okay, so there's a carbon there, there's a phosphate group on this, okay? So I'm not gonna do the phosphate group, right? This phosphate group, okay, might bind to what, guys? The 3rd carbon, right there, okay? So right there is what making this cyclic, okay? Like, dude, like a little ring here, right? So you see that, guys? That's what that makes what? Cyclic AMP. And this act is what, guys? A lot of times secondary messenger for the cell communication process, okay? How do they deactivate this? Oh yeah, the secondary messenger, you guys should stop, you know? They what? They break this bond, okay, make it into AMP again, Okay, but then what? Yeah, they are deactivated messengers now. Okay, so that's basically what cyclic versus like AMP is. So and yeah, so that's what it represents, guys. By the way, but yeah, I mean, definitely not the answer, right?
Yeah.
Yeah. Anything else? Any other questions, Mia? So guys, I mean, if you guys have a background, a little bit of chemistry, guys, please do not forget, okay? So it will help you guys, okay, for AP Bio as well, okay? Any other questions, Mia, guys, please? So like, I think I was referring to, guys, questions like number 20, okay? 20, 21, 22, 23. So that graph is not the Michaelis-Menten equation, the graph and equation that we talked about, okay? Because if you look at it, guys, I mean, it has a amount of product versus time, okay? So it's a little bit different than what you think it is, okay? The graph that we talked about.
We go over 22.
22? So you guys know the graph, guys, right? What is the most likely expression for the change in the slope of the between 3 and 5? So at like 3, amount the product increases, right? So so the rate of the reaction typically is what? So again, like if you look at the graph, which is also, one of the things that AP asks quite a few often, right? The graph looks like this. Okay, this is time. This is amount of product created. Okay, so now the rate. So you did AP Chem last year, right? Yeah.
This time.
Huh?
Regents time.
Okay, so how can you measure the rate of a reaction, guys? Yeah. What do you think? How can you measure the rate of a reaction? Andy?
How quickly the substrate disappears.
So equation, yes. Equation, how do you measure that? In this case, a product, right? So how do you measure in an equation, how do you get that?
Rate law?
What rate are we talking here? Differential, integration, integral, which one? Yeah, what do you think, Derek? I always just, what do you think? What I'm looking for equation, okay? What do you think?
The equation, I'm not sure.
AP Chem, AP Chem, AP Chem. AP Chem, so AP Chem, what do you think?
Just say integrated.
Yeah, let's set this straight, okay? This equation, right? You can't forget this, right? This is what? Differential rate law, right? Okay, so not to yeah. What does this equation tell you guys? What can change, what has an impact on the rate of the reaction, okay? So the reactant, okay, the concentration of the reactant has rate of impact on the rate, okay? And K is what, guys? Like I'm not gonna go arrange this equation, guys, I'm not sure. Energy of activation, temperature, and all have an impact on rate. So differential rate law tells you what, which are the factors that impact on the rate of reaction, okay? Which is not what I'm looking for, okay? So the rate, strictly from this equation perspective, is what, guys? Yeah, dude. How do I know the rate, guys? How fast the product is being produced, okay? How fast are they producing? Which is what, guys? Basically what? Slope of this line, slope of the curve, which is what? Changes of the concentration product over what? Change in time. That's the equation, okay? So how much of the product changes for a rate of amount of time? So if I'm choosing for time 3, right? So the slope is what? Increasing. Okay, which is what? Dude, product is being created. What about here? Yeah, what about here, guys? There's no product change, which is what? The rate is 0. You understand what I'm saying? So you gotta understand, guys, okay? So the rate is what, guys? How much product is being created for what? Given time. Okay, so if you look at the question, guys, okay? The question, guys, question is asking, which is the likely explanation for the change in the slope? So that slope is the freaking rate. Okay, so slope is a rate, right? So if you look at it, guys, change that, dude. Why did it stop? Why the rate stopped? Typically, what? I mean, dude, we didn't denature anything. Okay. Yeah, why did it stop? Maybe what? It ran out of the substrate, okay? Like, dude, it's gone. The substrate is gone, okay? It has, like, all the substrate probably has converted the product completely, or it's at equilibrium, okay? That's what it is, right? So I think, so it's not B, because like that's what I'm trying to say. This is not the Michaelis-Menten equation. Okay, that's what I'm trying to say here, guys. You know, that equation is what? Velocity versus what? The amount of substrate. That's not the correct dependent independent. Okay, so that's why I think the best answer is what, guys? Certainly very curious. I mean, D's not bad, but D's not a bad answer. But I think the best answer is what, guys? I think C is the most likely better answer, I think. So D is not incorrect. I tell you that because I know. But I think C is absolutely the better answer, okay? Any other question, let me know. Any other question, guys? Sorry, homework, guys? All right, so guys, move on to the next section, guys. Okay, next, I'll send you the reading, guys. Email through email, guys. Okay. So let's get started with the next chapter, guys. Okay. Yeah, who doesn't go to Jericho here, guys? Let me know. Jericho doesn't. Who doesn't go to Jericho? You guys all Jericho? Like, yeah. Do you guys all Jericho here? My goodness, what the hell happened? Okay. Yeah, I think some of the folks told me like they can't be here today or something. I don't know. Yeah, so but interesting. Yeah, what was that? Columbia Prep, right? Yeah. All right, so let's take a look at the next chapter, guys. So this chapter, guys, is cell organelles. And then the membrane and transport. Okay, so membrane and transport, right? So I'm gonna start off, guys, with the membrane and transport. Okay, so I'm gonna start with the membrane and transport, and then I mean this chapter, guys, I think it's really simple. Okay, for some. Okay. So let's take a look at it, guys. Put your phones away now, guys. You don't need your phones now. Okay? So let's talk about, guys, cell membrane now. So this one is a little bit more bearing on the AP level, okay? So which means you get to know a little more for the AP. Like the other stuff, man, the organelles, it's about the same level with like stuff that you learn, like 8th grade bio a little bit, guys. Like what does like Golgi apparatus, what do they do? You know, like AP level perspective. You know, like they're very simple. But this one now. Okay, so let's talk about this one a little bit, okay? So, cell membrane. So, first thing first, guys, right? What do they do? Cell membrane, guys. What's their job? What's their role, guys, in a cell? Okay, so cell membrane, guys. You gotta understand, guys, what they do. So, in AP bio question, they ask you, okay, what do they do? Okay, a role of the cell membrane. What do you think, guys? Cell membrane controls what comes in, what goes out of the cell. Okay, they're the gateways. Okay, typically like that, that's what they really ask you a lot. Like, oh yeah, what comes in, what goes out. Okay, so keep in mind, guys. By the way, okay, so in AP bio perspective, okay, so in terms of a cell cycle, okay, the cycle of a cell. So this is something that we'll discuss in maybe a few weeks later, okay? So in the cell cycle, it starts here, and this is what we call the G1 phase, and this is the S phase, this is G2 phase, this is the M phase and C phase, okay? That's the cell cycle, okay? Not that you have to really know this right now, guys, by the way, but you had to know this for the AP, SAT bio as well, right? If you took that before, right? This is called the GAP1 phase, which is the growth phase of the cell. Okay, S phase represents as synthesis, okay, which is like DNA replication process, DNA synthesis. And G2 is the second gap phase, mitosis, cytokinesis, the division of cytoplasm. So during G1 phase, which represents, guys, and sometimes they call it growth phase of the cell. So So when the cells grow, okay, and what happens, guys? Like cells grow, they start to what? Decide. We need to what? Divide. Okay, mitotic cell division here. So a lot of times, guys, just so you know, you have to understand what triggers the cell to divide. Okay, so what is this triggering process of the cell to divide? I mean, it could be many things. Okay, but AP Bio, very high level perspective is what? A lot of times you have to mention surface area to volume ratio of the cell. Okay, that's usually what they talk about. Okay, surface area of the cell membrane, okay, versus the volume of the cell. Okay, and then if the ratio gets too small, okay, the cell is what? Okay, it's a dude. We gotta divide. That's typically what triggers them. Okay, so now surface area represents what, guys? Again, how much of what? The cell membrane you have. Okay, which controls what? Which controls how much stuff comes in and goes out, right? What about the volume? So what's the volume of the cell represent? The cellular activities, okay? Such as what, guys? Like, oh yeah, like amount of metabolism that occurs within a cell. Okay, like it could be respiration process, protein synthesis, like all the stuff that happens within a cell, right? So volume. So keep in mind, guys, okay? Assuming the cell is spherical, right? Surface area is what, guys? The equation for the surface area is what? 4 pi r squared, right? What of the volume? 4/3 pi r cubed. And if you don't know this, guys, I mean, I don't know, man. Like, that's the surface area of the sphere. Okay, that's the volume of the sphere. You know what I'm saying, guys? I mean, if you don't know this, guys, I don't know, guys. So, dude, I'm not trying to make fun of you guys, but that's the equation, right? Yeah, this is the radius, right? As the cell gets bigger, which one goes up faster? Of course, this guy, because it's what? To the power of 3 versus 2. Do you understand, guys? So, as the cell grows and grows and grows, what happens, guys? There is a greater amount of metabolic activity, okay, versus what? How much of the cell membrane, activity increases. Okay, so what does it tell you is what, guys? If you look at it, there's a lot of activity going on, but what happens, guys? It's slow to ship out or bring stuff in. It's slower, okay, which slows down the entire process of the cell, which means what? It's ineffective, for the cell, which means what? I gotta divide. So imagine, guys, the perfect example is, guys, the city of Atlanta. Okay, Atlanta is where I went to college, guys, by the way. And you don't understand, man. I tell you, it's a little different than New York because this is the city of Atlanta. Okay, it's like a lot of places outside of New York, guys. The city looks like a little circle. Okay, they got 2 highways. Like one goes like this. Okay, this is like 75, this is 85. Okay, my school was like, I don't know, somewhere like here or something. Like I went to Emory, by the way. Okay, so like 2 highways. Like you don't understand, man. I tell you, whenever there is a rush hour, okay, they got only 2 highways. Okay, what do you think will happen? Like, dude, you sit on traffic for like 3 hours. If you live like here, you sit on the track for 3 hours, okay? So that's how bad the traffic is. The city grew really quickly, but there's not much roads, okay? That's exactly what's happening. So cell grows, what happens, guys? Yeah, there's a lot of people coming in, a lot of stuff happening in the cell, but what happens, guys? The cell membrane cannot keep up, okay? That's the reason why, oh, dude, like, yeah, would you want to live in a traffic where you have to sit on traffic for 3 hours, one way, to work here, guys? No way, okay? So anyway, so yeah, people. What happens to people, guys? They leave. Okay, so what do they have to do, guys? Either have a better infrastructure, or what? Maybe divide the city in half. I'm not sure, but typically, guys, that's basically one of the analogies I can give you guys. By the way, okay? So the whole moral of the story is what, guys? Cell membrane. What does it do, guys? I mean, yeah, it kind of controls what comes in and what goes out. Okay, so let's talk about the components of the cell. A cell membrane, I'm sorry. So, I mean, dude, like, I tell you, man, like, this is something you should already know. Cell membrane is what, guys? It's made up of phospholipids, okay? Phospholipids, right? So, you guys know the detail, like, little stuff, right? So, we talked about, guys, this is phospholipids, right? And also, guys, like, they call a phospholipid bilayer, okay? So which means, guys, there's 2 layers of this, okay? Like this. I mean, again, like this is more of a review for a lot of people, by the way. And then, so we discussed, guys, this portion outside, this is outside the cell, this is inside the cell, by the way. They're what? They're all what? Water. Okay, water, you know, is in and out, guys, by the way, okay? And now, as we discussed, guys, the hydrophilic portion, which is a phosphate, okay, facing the water, and what? There's a hydrophobic carbon tail is what? Facing the hydrophobic portion of the cell membrane, okay? So let's kind of talk about this a little bit, guys. Okay, cell membrane. Cell membrane is very, very important for the cell, by the way. If there's a puncture in the cell membrane, guys, what happens, guys? Cell dies. That's called the lysing of the cell. It's very, very important, guys. Okay, to have have a good cell membrane, by the way. Okay, like strong cell membrane. And just looking at the cell membrane by itself, okay. Let's just talk about this for now. So, if you look at this, guys, by the way, okay. So the temperature, right? So this guy is a very temperature sensitive, okay? I mean, again, it's for any molecule that you guys have, by the way, okay? Because, I mean, again, like how they're lined up is what? Just, you know, these two are attracted by what? Non-covalent bond, right? Which is what? Like very weak in body, right? And they call this what? Like fluid model, okay? Which is what? This like is like moving like a fluid, okay? It's like a water, like ripple. But the problem, not really a problem, but the issue here is, okay, when temperature increases, when temperature gets hotter, what happens? What do you think? Yeah, what happens typically, right? So typically what happens? Temperature gets hotter. Yeah, what do you think? Temperature gets hotter. What happens?
Things move faster.
Exactly. Yeah, things start to move faster. Okay. So they start to what? Like vibrate quicker. Okay. They start to have a little bit more rotational motions and stuff like that. And what happens, guys? They start to distance themselves away from each other. Okay. They start to drift apart from each other. Okay. So then, yeah, what do you think will happen? The gap will start to get larger. Okay. So when temperature goes up, what happens, guys? Temperature goes up. What happens, guys? They call fluidity increases, which means what? Like this is like they move a lot more. Okay, there's a greater amount of wave produced. Okay, so if temperature goes up and up, what happens, guys? There is what? This membrane will break. You know what I'm saying, guys? Here, like, dude, like they move too quickly. What happens, guys? They will break. So, what do you think what do you think something will help to reduce the fluidity of the membrane, okay, at a hotter temperature? For example, dude, like, you know, like, you will probably understand, you probably heard it, right? Like, dude, like, when temperature changes, right, your body cells change a little bit, too. Like, they have, like, a shedding season and stuff like that, right? Based on the fluctuating temperature, guys, like, you know, like, I don't know if you guys know, like, your hair, like, shed a little more, like, at a certain time of the year, and stuff like that, right? You know, I don't know if you guys, like, yeah, I mean, I don't know if you guys are aware of those things, by the way. So, your cells are temperature sensitive, by the way, okay? Even for like the polar bear, they always live in a cold area, but you know, like temperature has an impact on the cells as well, okay? So temperature's too hot, okay? So how do you think the cells will respond to that? What do you think? How do you reduce the fluidity when temperature rises, okay? One of the ways what? These hydrocarbon chains, okay, make them more saturated. Okay, so greater saturation. So greater saturation of the hydrocarbon chains. Okay, look at that. You gotta listen very carefully, guys, for now. So again, keep in mind, okay? So saturated means what, guys? These carbon chains are what, guys? Like straight line, like straight, okay? Like this is basically what? Straight carbon chains, okay? So if there's what, guys? All of these are straight chains. What happens, guys? You can pack them close together, which will what? Have the interaction becomes stronger. You understand, guys? So that's what happens when these hydrocarbon chains what? Saturated. But now, what happens if these tails are unsaturated? So which means what, guys? Okay, so unsaturated, which means like, yeah, what does it mean by unsaturated, guys? Double bond, exactly, right? Which means what? It produces a little kink, and what? You start to drift away. You understand, guys? So we talked about this, right? So now, because of this, what happens, guys? The other guys cannot get close. There's a gap now, okay? You understand what I'm saying? So which means what? Okay, so if there's unsaturated portion of the side hydrocarbons, what happens, guys? They're further apart. So greater the saturation of the hydrocarbon chains, what happens, guys? You can pack them close together, have a stronger interaction, and what? Decreases the fluidity of the cell membrane. So it's good to know, guys, okay? So yeah, increased saturation of the hydrocarbons. Okay, that's one of the ways to what? Their cells respond at higher temperature. Okay, it's not like something that can happen in one day. I mean, it's like, you know, gradual process, right? What else? Yeah, what else, guys? Temperature's still too hot. Yeah, what would you do? Their temperature's too hot, there's a gap there. Yeah, what would you do? You put something in there, okay? You put something in there that what? That stabilizes it. What is that, guys? Look up, what is that, guys? It'll be cholesterol. So they put a cholesterol molecule between the between what, guys? The phospholipids. Okay, so they put these guys in there. Okay, and what? So that what? It fills the gap between those two and what? Reduces the fluidity of the membrane. Okay, that's the other way to do so. Okay, cholesterol and what? Saturation reduces the fluidity of the membrane. Okay, that's for what? Responding to the high temperature. Okay, what about low temperature, guys? So okay, so here we go. That's all good. Right? Oh yeah. So if an AP question was asked you guys, which I don't think, I don't know. If they ask you guys, okay, so when temperature goes up, okay, what would the cell membrane? I mean, I've now seen that question like that on the AP, by the way. Okay, like fluid of the membrane, guys. I haven't seen that question, by the way. But your school exam question, who knows? You know, hey, what does it do? Yeah, cholesterol, amount of cholesterol should increase. Okay, and the saturation rate should go up. What about temperature goes down? Temperature goes down, okay, what happens, guys? Like, dude, they move slower, they're more like packed in, just fluidity what? Start to decrease. They're too rigid. They're not like, dude, they're not like bendable, like they're not like fluid, they're what? They're like packed in like an ice, okay? So what do you have to do, guys? You gotta make them into what? Fluid. So the reason why is that they got proteins on them, okay? So like, remember, have you heard, guys, fluid mosaic model? Hear that, guys? Okay. Like this is like, imagine, guys, these are like water. These are what, guys? Like the icebergs, ice cubes. Okay. They have to float around. Okay. They have to move around. They can't be stuck here. They can what? Like move around and stuff. But if the cell membrane, which is a phospholipids, if they're too rigid, what happens, guys? These guys cannot move around because they're too packed in together, right? So the question becomes, guys, here, okay? What can you do? Temperature goes too low, they have to increase the fluidity. Okay? Because what temperature goes down, fluidity goes down, it increases. What would you do? What is that? Completely opposite, right? You decrease the saturation rate. Okay? Which is what? You produce what? These like little kinks, right? So increase what? The decreased saturation. Okay? That's one of the ways you can do it. Okay, so which means what? Like if you look at it, guys, you put a little gap between those two. So this is basically what it looks like. Okay, so you put a little gap like this, which means what? There's a little gap, so they can't really get too close with the phospholipids, and basically what? Yeah, like that's something that can help them to maintain the fluidity of the membrane. Otherwise, they'll just freeze. What else, guys? Also the steroids. Steroid does help, guys, here. Not with the fluidity. But what, guys? The freezing point goes down, okay? So the cholesterol does help a little bit, guys, but what it does is what, guys? The cholesterol, so I don't think the cholesterol amount will increase because temperature is going down, but now what will happen is what? The cholesterol does assist, okay, to what, guys? Up to a certain degree, okay, to lower the freezing point of the cell membrane, okay? Just like what? Like you put salt in the water that reduces the freezing point. Same mechanism here, okay? And that's one of the things to things from percent, guys. Here, okay? So I don't know. Very unlikely your teacher will start to talk about like cholesterol and all that stuff, guys. By the way, but it's in scope, okay? Just so you know, for the cell membrane perspective. So you have to understand what they have and what it's composed of as well, okay? And because of, again, the cell membrane structure, right? If you look at this, right? So this is what, guys? Water. This is water as well. And now here is what we call the zone. I mean, so like, yeah, this is a zone of a hydrophobic. So this side is what? Inside the lipid membrane is what, guys? It's hydrophobic. Okay. So this is one of the stuff that we talked about, guys, by the way. Okay. So you probably heard the term, guys, the cell membrane is what? Semi-permeable. You heard that, right? So what can just go through and what cannot is something that one of the stuff that you have to know, right? I think we talked about this a little bit, right? What can just come in, guys? Small, tiny hydrophobic molecules, O2, like N2 gas, and also carbon dioxide gases. These guys can just come in through the membrane, okay? They can't stop this, okay? Because what? They're hydrophobic, nonpolar, and what? Very tiny, okay? But now, what cannot just come in? Because of what? There's a zone hydrophobic here. What cannot come in? No way the ions cannot come in. They will never be able to come in, okay? It doesn't matter how small they are, it doesn't matter. Ions will never come into the membrane, okay, by itself, like diffusion-wise. And what else, guys? So this is where you might, I might have mentioned this before, guys. Hydrophilic molecule, like water. Water itself is hydrophilic. I mean, hydro is a definition of water, by the way. Polar molecules. Even though they're very small, so this is where the problem is, guys. Your teacher could say, like, I had some issue with some of the teachers. I mean, some of the teachers like saying, some of the things that I don't think you should say. Water molecules, by the way, very they're very polar molecule. They can come in, very limited amount can come in, okay? So they have to, a lot of times water has to go through the protein channel called aquaporins. Okay, it's called aquaporins, which is a protein channel. So polar molecules are very limited, okay, a chance of coming in by itself through the cell membrane. Okay, they have to come through a protein channel like this, okay, which will assist the water to come in or make them come up a little faster. So that's one of the things that I'm saying, guys, hydrophobic zone. Okay, so because if these guys want to come in, what happens, guys? They have to come through what? Protein channels. Okay, that's one thing. That's one of the things you have to understand here. So far, guys, any question? I think it's straightforward, guys. Any questions now? So, guys, let's talk about guys, this proteins. Okay, let's talk about these guys. So proteins, there are at a high level 2 of the main proteins that you have to know in the cell membrane. Okay, we call this either integral proteins, okay, or sometimes there is what we call peripheral. Okay, peripheral. Peripheral proteins or integral proteins. Okay, integral protein. Okay, so protein is very, very important portion of the membrane itself, guys. By the way, so integral protein is where the protein is embedded into the cell membrane, like this guy. Okay, so it's like in there. Okay, penetrating into the hydrophobic. But now, integral protein can be what, guys? Also, what, guys? Integral protein can be transmembrane protein. Transmembrane is what, guys? Okay, so this is the outside and inside. They're open on both sides. You understand, guys? Those are called what we call transmembrane protein. So not all integral protein can be transmembrane, okay? Because some of the protein may be what, just sitting like this. Okay, so okay, it's like there, but it's not transmembrane. It's not connecting one end to the other end. Okay. And now you gotta understand this, by the way, because these transmembrane proteins and all those stuff. Yeah, so hear me out on this one, okay? So I draw the protein like a little circle. Okay, or do you a circle, guys? It depends, right? So we understand, okay, the proteins have what, guys? Proteins have hydrophobic zone, hydrophilic zone, and all that stuff, right? So now, if you look at it, this is a protein here. Okay, so you gotta understand. So this portion of the protein must be what? Hydrophilic. Okay, and this portion must be also what? Hydrophilic portion, hydrophilic portion, right? And now, okay, so, and then what? Then this portion of the protein must be what? Hydrophobic. So you gotta understand, guys, by the way, okay, that's what you know. So again, because this outside is water, inside is water, the middle portion is what, guys? Is zonal hydrophobic, right? Which means what, guys? That means so when you look at the structure of the protein itself, what happens, guys? Yeah, dude, you will see, like, as like they create this, like, you know, like that little like shapes that you guys know, tertiary structure proteins, right? So yeah, most of the amino acids you find here is what? Hydrophobic amino acids, okay? And what you find at the tip must be what, guys? Hydrophilic, or ionic, or acidic and basic, right? Amino acid at the tips, just so you know. Any questions, guys? Let me know. Questions, concerns? And guys, what about these the peripheral proteins, guys? By the way, so what is peripheral proteins, guys? So they are basically what? Like just sits like on the membrane. Like these are like what they call like peripheral proteins. Okay, they're not embedded into the membrane, but they're what? Just like sits on the cell membrane. Okay. So peripheral versus integral proteins. Okay. So now you gotta understand, guys. What are these roles? What are the roles of the protein on the membrane, guys? What do they do? What's their job as the these proteins on the membrane? So obviously, what, guys? Transport. One of them is what? Transport. So for example, guys, if sodium ion needs to come in, they have to come through a channel. Okay. So because why? They cannot just come into a cell through the membrane. Okay. Which means they, which is the phospholipid portion. They have to come through a channel. So they provide a channel for the transport of like glucose molecule, for example, guys, or even what? Water molecules, or what? Sodium ions, for example, guys, for even a little bit larger, and also what? Hydrophilic particles needs to come into a cell. Okay? So like, given the fact that, guys, like these control what comes in and out, okay? Imagine, guys, there's a lot more activity going on because the cells get larger, but these channels are what? Limited. Like you probably heard, right? A couple of months ago, right? What do they have, guys? The Suez Canal, you know, like they got backed up, you know? What happens, guys? They got what? Your product that you bought probably was backed up also as well, right? Because why? Transport only can happen what? Through these little channels for some of the particles. So that's one, right? What happens, guys? So these enzymes can act also as well as an enzyme. They can act as an enzyme. Okay, so these proteins on the membrane can act as an enzyme. So for example, one of the ones you have to know. You have to, like, again, like this will come, I don't know, probably, guys, maybe next few weeks. There's an enzyme called, as an example, adenylyl cyclase. It's called adenylyl cyclase. Okay, it's on the membrane of the membrane. Enzyme is on the membrane. The protein is on the membrane. Okay, you gotta memorize, by the way, adenylyl cyclase. Okay, this is cell communication, by the way. So what they do is basically what? They convert ATP into cyclic AMP, okay, for the cell communication perspective, okay? So ATP, adenosine triphosphate, right? They take out the two phosphates and what? Turn it into what? AMP, okay? Make them into cyclic AMP, okay? That's what the job of one of these enzymes on the membrane, which, again, one of the best one that I can give you guys is this guy. The examples you have to know. This is for the chapter of the communication, by the way. Okay, cycling, we talked about what these are, what this is, and stuff like that, right? So that's one of those. So these enzymes, these proteins can act as an enzyme as well, right? And then also, guys, the other role of this is what, guys? So again, it's a cell recognition. So which means, guys, so yeah, if this is a protein, right? So proteins, a lot of proteins, by the way, they're what they call, guys, glycoproteins. Okay, they got what? Like sugar molecule there. Okay, so again, the cells, right? Like cell recognizing. So yeah, think about it, guys. Okay, now what do they do? Like when you're getting, like, not you guys, when someone is getting like a transplant, right? Like kidney transplant and stuff like that. Yeah, what do they do? Yeah, what do they check, guys? Yeah.
Blood sample, like immunosuppressants. Actually, they usually prescribe like immunosuppressants, right?
Yeah, but what do they do that though? What do they provide them, like the suppressant when someone gets a transplant? Because why? Yeah.
You might have the same antigens in your body can like attack it.
So your immune system, what do you do? What does the immune system do, guys? they go and check, okay, if the cell belongs in your body. Okay, they're what, guys? They have every cell in your body has a protein tag, okay, saying that what, this is your cell, it belongs in your body. If they do not have that tag, okay, or missing the tag, have wrong tag, different tag, what happens, guys? The immune system will attack it. Okay, what is that freaking tag, guys? Protein. Okay, like a protein like this. Okay, so those are the protein that's found on the cell membrane. Okay, which can be what, guys? Which can act as what? Cell recognition. Okay, so that's the reason why. Okay, so can you give like your not you? Can someone just give like an organ to the other person? Go, oh yeah, I heard that you have a bag. Here you go. Can you do that, guys? You can't. Cause what? Like, dude, like, yeah, recording. So you can't because what? Like, yeah, like if you get it, like they can start, your immune system can start to attack, even the same blood type and stuff like that, right? So that's why, like, all these every cell in your body got these what? These recognition sites. Okay. So and then it's one of the things that the cells look at a lot, guys, for the recognition perspective. Now a lot of times, guys, the carbohydrates on them is very unique for each cells and also for the personnel. And like the other stuff, man, like they act as an anchorage, okay, anchor down. So which means, guys, like, okay, so for example, guys, like there is something called ECM. Okay, I don't know if you have the time to talk about it today, guys, by the way. It's called extracellular matrix, by the way. Okay, so it kind of hooks stuff onto ECM portion of it, guys, but it's like not so important right now. And also, guys, 5, the cell communication. I'm sorry, cell communication, yes. So, which means, guys, I mean, it acts as a, like, which means that these proteins, guys, by the way, there's these receptor proteins which will kind of be able to sense signals per se, guys. Okay? So those are some of the high-level function of these proteins, by the way. Okay? So it's good to know, guys. I mean, probably we'll have in the reading, guys, as well. So I can't just talk about this all day. Okay. So one of the things I want to talk about right now, guys. This is, I mean, they don't talk about, guys, oh, what's the role of the protein? I mean, maybe your teacher will be. Okay. On the AP. Okay. The main thing they will talk about the cell membrane is transport. Okay, that's the main thing to talk about. Okay, transport is the main thing. Okay, not like, okay, so like little kink on this, like, oh yeah, like cholesterol. I don't know. That is not the favorite thing they talk about, guys. The favorite thing to talk about is, okay, transport. What comes in and out. So let's talk about the very high level, simple stuff. So this is where, guys, you guys have to know a little more for AP. Okay, compared to the easy stuff that you probably learned, guys, by the way. There's an equation involved here as well. So this is where, a little bit of math gets involved in bio, okay? You think about it, right? Let's talk about this. So, one of the things you might already know, okay? Let's talk about this. You got distilled water. I'm gonna throw a question straight up, guys, okay? Distilled water. What's a distilled water, by the way? Yeah, what's distilled water? Here. You can kind of say what it's got only what it's got minimal stuff in it. It probably has what, like, it's probably doesn't have any minerals in there. It probably has what, guys? Some of the H2O, because they sort of distill, like heat it up, okay, and then condense it. That's what distillation is. But so yeah, it has a minimal stuff in there, okay? It might still have some gas molecules in there, by the way, but very minimal stuff, isn't it? So basically, guys, there's like, just like what he said, like, let's assume there's nothing in there, okay? And now you put a cell in there, okay, into a pure water, okay, or distilled water, whatever we call this. What will happen? That's one of the things they ask a lot. Okay, the transport questions, right? Like, you put that guy in there, what happens? Okay, that's one of the stuff you have to understand. So now, within the cell, okay, so there's some stuff in there, right? They got, like, some of the ions and some of these other stuff in there, guys, and also the membrane itself is what else? Membrane itself is basically also what? They call this semi-permeable. Which means what? Yeah, what does it mean, guys? something can pass through, something will not be able to, okay? Such as what? As we talked about, very tiny small molecules like N2 gas, O2 gas, and CO2, they can pass the membrane. Other stuff as well. Very hard time passing the membrane, okay? That's called a semi-permeability of the membrane, right? So then, if you put this in water, what will happen is a question, okay? So you put this guy in water, okay, in pure water, right? What you will see is a question, right? So this is, guys, so one thing is what? So one thing to understand is this. One thing that can happen is what, guys? So these particles, if they can, what happens if they want to go out, okay? So the reason is, this is a little hard to explain, guys, by the way, like why they want to go out, okay? So the reason is entropy. Okay, entropy is the reason, okay? So, because, think about it, guys, okay? So if right now, nicely, all the particles are inside this enclosed space, okay? Which is what? Relatively low entropy, okay? But the universal entropy tends to increase, which means what? They would like to what? Go everywhere else, okay? So that's due to entropy, right? So for example, guys, I don't know, if we have a train cart, okay? There's a little like room or whatever, guys, whatever, okay? And in the room, guys, you put a little bit of a container with flies, okay? Like you put a little flies into the container, okay? You let it open. You let the doors open. What do you think will happen? Do you think they're gonna stay there? What do you think? No, they're gonna go all over the place. You understand what I'm saying? So, why? Entropy. So entropy, like the molecules are not like flies. Okay, flies might communicate also. They might like, congregate in a one spot because of the mating and all that stuff. But again, these are a little more random, okay? They want to go out. But the question is what, guys? Can they go out is the question, right? So can they go out is a question, which means what? A lot of times because of a semi-permeability membrane, they cannot. They cannot, okay? Like very small things such as CO2 can go out, O2 can go out, N2 can go out. But what? Like other stuff, they cannot, okay? So then what will happen, guys? Like, what will happen? Then they just give up? What do you think, guys? No. Water is going to come in. Okay, the water will come in instead. Okay? Instead of these particles going out, the water is going to come in. Okay, that's what's going to happen. So you got to understand why. Okay, this is where, guys. Okay, so you probably already know some of these things, guys. Okay, so in bio they call this what? The outside water is what they call. I'm sorry. So outside water is what they call as what? So inside here, there's more stuff in it. Okay, what we call this what, guys? Hypertonic. Okay, there's more crap in there, okay? Greater osmolarity. Outside is what, guys? Hypotonic, right? So the water flows in from the hypo to hyper. That's one of the stuff, guys, they probably teach you a lot in like the regular bio classes, okay? But in AP Bio, you have to know why. Why is the water going in? So water moving in and out of the cell membrane is called the osmosis. So why is the water going in? Listen carefully now, okay? This is a question you have to know. It has to do with something we call water potential. Okay, this is the equation that you have to go for AP Bio. They will have this equation, you gotta solve it. The equation goes, water potential is equal to, the equation is, this is that's a sign, okay? Is equal to negative I times MRT. That's the equation for water potential. This is like, dude, I tell you, sometimes they call this water potential, sometimes they call this, guys, osmotic pressure, the equation, guys, right? So it is a pressure, yes, okay? Because the unit of this is in bar, by the way, okay?
Is that times MRT or minus MRT?
Times. So yeah, you've probably seen this equation somewhere, right? Like, you've probably seen equation like this. I mean, depends on when you're at, guys. What do we call this, guys? This is a Van Hoff factor. Okay, AP Bio, they don't want to play around with this too much, guys, by the way. But you should kind of have understand what that is, okay? So water potential is equal to, guys, minus times I, this is called a Van Hoff factor, which I don't think for AP Bio you have to be concerned about this one too much, guys. That's called a concentration molarity. This is a constant, okay? I mean, so do you guys remember from chem, guys, last year, the PV&RT equation? Right there, R value right there, same R value, okay? But, listen carefully, okay? Yeah, what kind of R value that you use from chem, right? What does this depend on? What do you think? this R value depends on what? Whether you use 0.0821 or like 80, around 80 or like 8.314, depends on what? The pressure. Okay? If the pressure is ATM, use what? 0.0821. That's the value bar. Just so you know. But now, in bio, okay, the pressure is given as bar. Okay, like 1 bar, 2 bar, 3 bar. Like they use bar as pressure. Why? I have no freaking clue. Okay, I don't like it. They use bar. So, yeah, keep in mind, okay? To give understanding what bar is, 1 ATM is what? 101.3 kilopascal. That's conversion. Kilopascal is the SI unit for pressure. Okay, like what, meters or something, you know? Like the West, like the kilogram, you know? 1 bar is equal to 100 kilopascal. Okay, so you can kind of see the resemblance here, right? Like, dude, like you see that, guys? It's about the same. Okay, about the same. Okay. So 1 ATM is 101.3. 1 is what? 1 bar is what? 100 kilopascal. So it's about the same level. Okay. But now the R value is a little different, guys. I think if I'm not mistaken, I said 0.083 or something. Okay. That, again, like that, I don't memorize the R value for all the things. Okay, by the way. So it's about the same. Okay, that's R value bar. Okay, but depends on the pressure. A lot of times, guys, in bio they will use bar. I'm for sure. Okay. Temperature. This is temperature, guys. Temperature must be in Kelvin. Okay, like 298, for example, guys. Plus two. You gotta add the temperature Celsius plus 273. Okay. So that's the equation, guys, for the water potential. Okay. So let's take a look at this. Okay. So for distilled water, right? So what? Yeah. So think about it, guys. What would the potential for water potential, guys? Okay, or osmotic pressure for distilled water? Yeah, distilled water. Right? The equation is this, right? Yeah. So what's the concentration of distilled water? Now what do you think? It'd be what? 0. You understand, guys? If it's 0, there's a minus sign there, which means what? Still what? This will be a 0 bar. Okay. Distilled water is 0 bar as the what? Osmotic pressure. Yeah. What about for the cell? I'm sorry, there's a little mess here, but what about the cell? So the outside is 0 bar. What about the cell? Do they have some stuff in the cell? Yes, they do, right? So this will be non-zero value. There's a minus sign there, which means what, guys? Like, I mean, so for example, this would be what? Like negative 0.3 bar, for example. You understand, guys? That would be the osmotic pressure within the cell. That's for what, guys? The pure water. So now, what will happen? Yeah, so this one is a high pressure, that's a low pressure, right? What's gonna happen is the question, right? So, yeah, so have you heard, guys? Make sure to know the equation, by the way. So you gotta memorize this, by the way. Yeah, have you heard, guys, like, you know, like, nice. So if you look at it, guys, right? So this is like the United States. I'm sorry, guys, the whatever. Because this is the United States, right? We're like here. So like, have you heard this, guys? By the way, the high pressure from the north, like, you know, the wind blows from high to low pressure. Have you heard that guy? Yeah, dude, like, unless it's on the rock, guys. I mean, the wind blows from the high to low pressure. You understand, guys? So when it's really cold, like, if your window facing north, they get all the cold water, cold air. Okay, so yeah, air is a fluid as well. Okay, they go from the high to low pressure. Okay, high pressure to low pressure. Okay, yeah. What do you think? Yeah, what do you think will happen here? Water is a fluid. Air is a fluid. Okay, what do you think, guys? The water will flow from the high osmotic pressure to low pressure. Do you understand, guys? So yeah, what will happen, guys? What do you think? The water will go from what? 0 bar to what? Into what? Into this. Do you understand, guys? So that's what we call this what? We call this osmosis. Yeah.
If pressure is like particle interactions, right? What does a negative pressure even mean?
That's a good question. That's a good question right there. Like, you guys should have the same question, right? So pressure, right? Like pressure, like as I said, right? This sometimes they call this osmotic potential. Okay, water potential energy. Sometimes it's called this equation as osmotic pressure. So whenever they talk about a potential energy, okay, like, or potential, like, you know, energy or whatever, energy perspective, right? they need to have a standard value, okay? Like the starting point, okay? So one of the things that I can tell you, okay? Because like, for example, right? Like, you know, like one of the examples I can give you is like smartness, okay? It's like, okay, so how do you measure smartness of a person, right? So, you know, even from my perspective, like, dude, I've been doing this for almost 15 years now, okay? I'm teaching 15, 16 years now, okay? So what am my standard is, so if you guys got like, you know, 10 questions right out of like 15, okay? Then like, what is my standard knowing like this kid is not, you know, this kid is not smart, that kid is smart, like, what is my standard, okay? So then I take one of my best students that I taught for the past 15 years, and then compare that, your grade to that person's grade. And okay, if you're about that range, okay, you're smart. If not, you know, and if it's higher, then yeah, it's a smart kid, right? That's my standard, okay? You gotta have that little what? Like that standard, okay? So that standard a lot of times is set as 0, okay? So the example is, okay, one of the things, so like, it's hard to kind of, you know, give you an example of this, okay? Is, I don't know if you recall this, by the way, like electron in the atom kind of thing. I don't know, potential is electron in the atom. Did you do the AP Chem last year?
Yeah.
So, okay, so the Bohr's equation, right? So it's a good question, by the way, okay? So N1, okay, N2, this is the principal energy level, right? N to infinity. Okay, so this is like principal energy, how far the electron is within the atom, okay? So if electron is on the first level, what is its potential energy? It's negative 13.6 eV. That's a negative sign right there, again, okay? If electron is the highest level, what happens? There's what potential energy is, 0 eV, right there, 0 eV, okay? So you've probably seen this somewhere in the AP Chem as well, okay? Like negative 13.6, what the hell? Where did you get a negative sign, okay? Because why? They set the standard as a potential energy electron when it's what? Really far away, okay? So it's the same logic, okay? Which means if there's nothing in there, that's highest point, okay? And then when there's more stuff in there, as the pressure gets lower and lower, they put a negative sign there. Because it's easier to go that route as setting a standard at what, 0, and there's nothing there. So it'll be either 0 or negative values. That's basically how they came up with the equation. And that's a lot of times what it occurs as a potential energy equation per se, okay? And you probably, when you take physics next year, same thing applies for the gravitational potential energy as well. Okay, 0 is highest gravitational potential energy, and it goes down, okay? So that's a good I don't know if I fully explained to you, but that's a very good question to ask, okay? Why there's a negative sign, okay? So again, so water will flow in from a high to low, okay, by osmosis, okay? Until when? Until when, guys? Until when water will flow in, guys, by osmosis? Until the pressure becomes equal. You know what I'm saying, guys? Yeah, pressure becomes equal. So now, that's what AP will say. Yeah, will it ever be equal? When can this be 0? There's some stuff in there, right? When can this 0? When what? A lot of water goes in, okay? So yeah, think about it, guys. Water is gonna go in, water is gonna go in, and what happens? Cell is getting bigger and bigger and bigger, okay? Can they withstand this? What we think? They can't. What happens, guys? The cell will burst. They die, okay? Because what? Too much water is going in. You understand what I'm saying? Okay, so have you ever heard, guys? Like for example, guys, if you put a seawater fish, okay, like you caught a fish, right? And then you put them in like, oh yeah, I'm gonna make it live. I'm gonna maybe raise it, and then put it into fresh water in your tank or something. Would it survive? Very unlikely. You know what I'm saying? Because if you put a saltwater fish, their cell is filled up with salt. Put them into a 0 bar osmotic pressure environment. What happens, guys? Water's gonna go in, the cells will die. You know what I'm saying here? Okay. So that's the understanding of what we call, guys, osmosis. Okay. So just letting you know. So basically, guys, I mean, I don't know why, guys, a lot of bio kits, even for the AP kits, guys, they have a hard time understanding this. They get confused, I think. Okay, so you know. Why? I have no clue, because you know. But when this go in, and you will never bother the valley, by the way. Okay, when the water goes in, what happens, guys? They'll lyse. Okay. But now, okay. So will any cell die in a case like this, guys? What do you think? Can a cell fight this? Is there a way that the cell can fight this off? Yes. Okay. With such as what, guys?
some cells have a contractile vacuole, okay? Some cells, I mean, I'm sure you ever heard that, guys. Contractile vacuole, which is what? They pump the excess water out. Like paramecium, they have a vacuole, guys. Like they pump out the water, you know, so they pump it out so what? To survive. There's a mechanism where they can fight this off, okay? The question becomes, guys, so this is one of the stuff they ask on the AP, guys, okay? What about for the plant cells? What about for the plant cell, guys? So a plant cell, okay? So this is a water potential. So this is the equation you might have to know. This is AP, okay? This is strictly AP right now. And also for you guys too, like Olympiad stuff. This is the overall potential, okay? And this is what they call, this is basically, guys, sometimes they call it solute potential. That's what S stands for, okay? Which refers to that equation right there, okay? Negative MRT. And this is called, guys, a pressure potential. Pressure potential. So guys, I mean, again, like, okay, so you probably went to a market, okay? You probably went to a market with your mom and or your dad, whatever, okay? Or you're by yourself or something, I don't know. And then you're trying to buy some vegetables, okay? What do they do? Yeah, if you go buy vegetables, guys, typically what they do is what? They have like water coming from like a machine, like spraying water on the vegetables, or they got some kind of water there, okay? To make them look fresh, I guess, I'm not sure, okay? So why can plants withstand, okay, like these pure water? Why? Like, you know, why can they do that? Because why? They got something we call a pressure potential on the plant cell. So like, how does that work? So let's take a look at this, okay? So the question is asking, guys, by the way, your question is asking, okay, so you're putting a plant cell in a 0 bar osmotic pressure environment, okay? And now, for example, guys, okay, so within a plant cell, they have what? They have a negative like 1.5 bar, okay, as the solute potential within a cell, negative MRT, okay? How can they withstand this? Something we call what? A pressure potential. Okay, where does the pressure potential come from, guys? The cell wall prevents the cell from lysing. So basically what happens, what? This is a plant cell, right? So basically what happens is, guys, plant cells have what? Yeah, plant cells have what? Yeah, what do they have, guys? They got what? Cell wall. Okay, they got cell wall. So many of these cells that lives in water, unicellular cells also as well, guys, they got cell wall, okay? So cell wall of the plant is what? Made up of cellulose. Very durable material, right? So some of the fungi have cell wall, some of the prokaryotic cells have a cell wall as well, guys, by the way, okay? What does it do is a question to ask yourself, okay? There's a little casing around it, right? Yeah. So there's cell membrane here. Cell membrane is within. Cell membrane is inside the cell wall, okay? So what the hell happens, guys? Water goes in, okay? Because why? Okay, so right now it's 0 bar outside, negative 1.5 bar, negative 1.5 bar is a solute potential within the cell. What happens, guys? But the cell wall helps them what? Helps them to withstand the pressure, okay? That's called, what do we call this, guys? Pressure potential of cell wall, okay? Of what? 1.5 bar. They asked this on the AP. Yeah, they asked this on the AP. Oh yeah, calculate the pressure potential. Okay, or if the outside pressure is 1, outside is 1 bar, 0 bar, solute potential, we have to calculate this. Okay, they're asking what the hell is this now? They asked this question because we know. So there are more stuff, but at least you have to know the solute potential, which is this one, okay? The osmotic potential, whatever. And also the pressure potential, which is positive value, exerted from the cell wall, okay? And then, which will be what? Okay, 0, which means what? It's fine, okay? And then when they swell up, right? They swell up, okay? That's called what? They're saying, oh, it's being turgid. They swell up, okay? And now, this is when, okay, there's a lot of water in these, you know, like produced when you guys go to like a market, and that's when they look fresh, okay? Like, oh, it's so hard, it's so fresh, like, you know, like that's what happens, okay? But now when the water comes out of these plants, what happens, guys? They start to shrivel, okay? And that's when they look like very, like, you know, not fresh and stuff. So that's why they start putting water there to make them a little more fresh, and then the cells becomes turgid, okay? I tell you guys, like, out of all the transport stuff you have to know, they will ask this question the most, and this is something I think, I don't know, man, like this is where students get confused a lot, because they got the math part, you gotta calculate this, okay? Like they will ask for the calculation portion of it. So this is one of the question, guys, one of the portion of the AP where you know, like you gotta know the equation, like chemistry guys, like PBRT equation guys, you gotta know your equation in order to do. Okay? Any questions? Let me know, guys. Questions? Let me know, guys. All right, so let's talk about, guys, like very simple transport stuff, guys, like that. Also, so the main class of, guys, I mean, transport is passive transport. Okay, here's an active. Very short on this one, guys. Active and passive. Okay, how do you decide what's passive, guys? Active is what, guys? They use ATP. Going against the concentration gradient. Okay. Passive is what, guys? No ATP. No energy is used. Okay. So what's the examples of passive versus active? What's some examples of passive, guys? Is what? Diffusion. They have diffusion, facilitated diffusion. Okay, and also what, guys? Osmosis. This is what you have to know. Okay, these are examples of passive transport. Diffusion, facilitated diffusion, and also osmosis. Okay, ATP is what? Active transport, right? It's not active. Active transport is ATP, by the way. Okay, so some of the examples is what, guys? Something you have to know is something called co-transport. Okay, like also what? Like what? So the phagocytosis, you know, like pinocytosis, you know, like cell-mediated, like whatever. So these are the examples of what active transport. You got to pump it out. You know, so one of the ones you have to know, guys, as well, like sodium, potassium pump. Okay. Well, that's one of the stuff that's examples of an active transport, by the way, okay? Which requires energy, which is ATP, to what? To transport something against the concentration gradient. So what does that mean, guys? So the active transfer is very simple, okay? Like it's one of the simple thing, guys, by the way. So let's talk about, guys. Okay, so you guys should know that what the hell is this, right? Like you guys understand what it is, right? So let me give an example, guys, what co-transport example is. Okay. So co-transport example is basically, guys, I mean, co means what? Yeah, like it's like, you know, something together, right? So one of the examples they like to give a lot, as a not a lot, but as a co-transport example, which you have to know, is that glucose transportation. Okay, after you eat like something sugar stuff and your body is filled up with glucose, okay, in the small intestine, how do they bring it into your cells, right? Like, you know, in the small intestine, from the lumen of the small intestine into the cells, right? Epithelial cells, by the way. So how they do that, guys, is this. Okay, so you have a high glucose concentration. And then you have relatively low glucose concentration. So typically, yeah, what's an active transport, guys? You want to bring something from a low to high. Okay, so that goes against the gradient. Okay, against what? The concentration gradient. You understand what I'm saying, guys? This doesn't happen. Okay, by passive. Doesn't happen by passive. Okay, because like you want to shove it in. There's a lot. Okay, you got to shove these guys in. Okay, back into a cell. Okay, or into a cell. It's only what? From a low to high. This is what we call, guys, what? It's going against the concentration gradient. Okay, which means what? This has to be at the what? Expense of ATP. How do they do it? So this is an example, right? How does these cells in your body bring these guys in? How do they do it, guys? So that's example of co-transport. So one of the ways to do it is this. So what they do is they got a sodium pump, okay, sodium Na plus pump, which they move, okay, sodium outside the cell. Okay, so there's a greater concentration of sodium, high concentration outside the cell, low concentration inside the cell. Okay, they push them out, and then basically what happens, what, guys? What will happen is the sodium will bind to glucose. Okay, they will create a bond with each other. Okay, and then what happens, guys? There is a little channel, protein channel. Okay, that will what? Sodium, okay, if you look at sodium perspective, what happens, guys? Sodium has what? This is a high sodium, low Sodium. Okay, sodium can come in. You understand what I'm saying? Okay? But what happens, guys? It will tag glucose along with it. Okay? That's how your epithelial cells bring in what? Sodium into the cell. Okay? And that's an example of what we call guys what? Co-transport, which is in with AP. Okay? Any questions? Let me know, guys. Questions, concerns? So, like, think about it, guys. Okay, so you probably hear, right? Oh yeah, like you guys have to have your minerals, okay, electrolytes in your body. Okay, you guys a lot of times, guys, you guys need like sodium ion, potassium ion, calcium ion in your body, guys. Because why? They have multiple roles in your body. Okay, like stuff like this. That's example of what, guys? Active transport. Which means what? So, do you know, guys? What they ask you here? Do you know what they ask you guys, by the way? Like, so this is a question, right? Just Constantine just explained to you, right? They ask you, okay, which process directly, okay, gives the energy for glucose to come into a cell? Yeah, what would the answer? Directly provides the energy to have the glucose come from the low to high concentration. What would be the answer? Huh? Yeah, right there. ATP, yeah, ATP, right? Is it ATP? What do you think? Yeah, so 90% of students will choose ATP. Okay? Yeah, ATP provides energy, but you gotta understand the term direct. Okay? So, like, for example, guys, right? Your dad like got your birthday gift. Okay? Your dad has to go to work early. Okay? And then he comes home late. Okay? So your dad gives your mom the gift, and your mom gives to you. Okay? Who directly gave you the gift? Exactly. Okay? But who actually got it for them? Your dad. But the question on the AP, they play with words and, you know, like the terminologies here, okay? The question asks, what directly provides the energy for the glucose to come into a cell? This guy's indirect. ATP's indirect, okay? Is what? The concentration gradient of sodium ion is what providing the energy. That's AP question, okay? So, I tell you guys, AP Bio, guys, you can be a very good at bio overall, but the wording they play around with, guys, they're just, you gotta be good at reading, by the way, like an interpretation. So you're right, indirectly, ATP provides them with all the energy for the process. But directly, it's what? It's this guy. Because what? That's what the definition of co-transport is, okay? And like, dude, I have to explain this guy. You guys know these guys? Yeah, the diffusion, right? You guys, you know. So I like save time, guys. Yeah. So these guys, show you guys where. I have notes here. So, okay. Any questions, guys? You know. So guys, yeah, so again, like, I'm not done with this chapter, guys, by the way, so I don't think, I don't know, we can go any further. Because and then after this, I mean, so the transfer stuff, guys, I mean, it's not a big deal. I mean, they have questions, yeah, they do. Just have to know what it is and the definitions and all that stuff, guys, okay? And then just terms, and that's about it. The structure, they don't really talk about that much. But one last thing I'm talking about, guys, is this. So, I want to talk about, guys, the ion gated channels. That's one of the last things I'll talk about in a transport chapter. Okay, so next week probably I'm gonna talk about organelles, and that's about it for this chapter. And then the week after we'll probably talk about, guys, the respiration and photosynthesis together, and then cell cycle and communication and all. Okay, and genetics. The time will last for us. So I'm gonna channel. What the hell is this? So a lot of times, man, like the questions that you get a lot, guys. So one of the examples I'm gonna provide you guys is the mitochondria or chloroplasts, okay? The way they produce ATP. So guys, I mean, so I'm gonna be very simple here, okay? So they produce the mass amount of ATP is what they call oxidative phosphorylation. Okay, so whatever the terminology we'll discuss later, guys. By the way, so basically what they do is that they pump out H pluses because it's called protons, right? They pump out these pluses by active transport, and then they have. I mean, if you ever heard this, guys, by the way, they call it ATP synthase. Okay, I mean, if you took some kind of bio, you probably heard this, guys, by the way. ATP synthase. Okay, this guy opens up. Okay, H plus comes in. Okay, so the reason I'm giving this example, okay, is okay. So there's a question. They pump out these pluses. There's a greater concentration of H plus outside. Okay, it follows the concentration gradient. Okay, chemical gradient. Chemical gradient means what, guys? There's more of this guy compared to the inside. Okay, they just come in. Okay. That's that's the example of what? That's the example of facilitated diffusion, right? Like coming through a channel, okay? But now, they don't say H plus come in using the chemical gradient. They don't say that, okay? So if this is just like, you know, whatever, like H here, okay? You can call that what? Oh, it's following a chemical gradient, high to low, and comes in as a what? A facilitated diffusion, okay? But they don't say that. They call this what? Electrochemical gradient. They call this electrochemical. What the hell is electro? What's electro here? What do you think, guys? We call this electro because what? These are charged particles, okay? They're charged particles. They're plus particles, okay? So not only there's what? Less amount of chemical concentration of H plus, but also what, guys? They have what? Greater voltage, okay? Like electro voltage, positive charges here. So there's what? Higher potential outside and lower potential inside, okay? So not only, guys, okay, they're following the typical diffusion process, right? But they're also what, guys? Following the potential. So high potential means what? There's more positive charges. There's what? There's more negative charges. So these positive charges will come in. Okay, so they call this in AP, guys, electrochemical gradient, okay, because of what? Dealing with the positive charges as a particle that's coming in and like being transported. Okay, that's one of the things that I'll talk about, guys. So these are what they call, guys, ion gated channels. Okay, I mean sometimes they call this voltage gated channels, by the way. Okay, so some kind of voltage triggers them to open and just come in and stuff like that. But again. Yeah, we'll discuss this more, guys, next week. So your homework, guys, I'm gonna send you the reading. That's it. Okay. So there's more organelle part that we'll discuss, and then that'll be it, guys. Good reading wise. Okay. So you guys can, I don't know, you guys maybe, yeah, listen, you guys may be able to do the homework, guys. Okay. So the reading, yeah, you guys for the whatever that's assigned, and try. Okay. And then let's go. All right, guys. I'll talk to you guys next week.